Hello, everyone. My name is Mats Hansen. I'm the CEO of the Swedish nanomedical company Spago Nanomedical. I'm going to share some slides today with you to explain what we do and to touch upon our exciting projects within precision diagnostics and therapy of cancer and other life-threatening diseases. So Spago Nanomedical is based on a proprietary platform technology which positions us in a way that we can use precision diagnostics and therapy based on this platform, which is now clinically validated in breast cancer patients. So using our unique platform, we are using functional nanoparticles that can be used to selectively accumulate in tumors and other tissues with fenestrated capillary vasculature. So we call this physiological targeting by means of the enhanced permeability and retention effect that is typical for many solid tumors and some other diseases or conditions. This is now clinically validated. We have used our diagnostic functional nanoparticles in patients with breast cancer. Our product candidate SN132D is a tumor-selective contrast agent for MRI, which has been shown now to in a very nice way and selective way light up breast cancer tumors in humans. With this, we open up the use for this platform also for therapeutic use. And as you can see in the image to the right, we have shown also in animal models with our therapeutic candidate drug, LU177SN201, that we get good accumulation in tumor, in this case, in the mouse tumor colorectal cancer. So with this, we open up for use of our platform material, both for cancer diagnostics and therapy. Pipeline is based on two projects or two programs. SpagoPix, which is our imaging diagnostic for MRI of solid tumors and other conditions. It is now completing the first phase 1 trial, where we got images, very nice looking images in breast cancer patients. We did start to look into pancreas cancer as well, but we are now abandoning that and focusing the continued development of this program into endometriosis, where we just recently announced that we are starting a phase 2 trial. So the lead candidate here, SN132D, is a lesion-selective contrast agent for precision MRI. And as I said, we do have clinical proof of concept now, or proof of principle in breast cancer, and that validates the use of our entire platform, basically, in humans. And this opens up, of course, for therapeutic use as well, by means of our other program, Tumorad, where we are developing LU177SN201 as our candidate drug for tumor therapy. And this is a really unique and IND-ready radionuclide therapy with broad potential, both as monotherapy in solid tumor, but perhaps even more interesting, with a mechanism supporting combination treatment with, for instance, checkpoint inhibitors or other targeted treatments. I'm going to start to talk a little bit more in detail about SpagoPix and just explaining early on what these particles are about. We are working with a concept based on functional nanoparticles, which means that the particles themselves are able to do what we want them to do in the tumor, in the tumors. So they are based on a polymer loaded with manganese, which is a splendid element for contrast with MRI. And these particles are designed in a way that they selectively accumulate in tumors, which means that we can provides for better precision imaging and less background compared to what is seen today with current contrast agents. The clinical concept is quite simple. As I said, we're using manganese, which is an essential element and can be safely used in humans. It's paramagnetic, which means that it provides excellent contrast in MRI. The nanoparticles, as I said, are functional in the way that we don't need to release anything into the tumors. It's just their sheer presence in the tumor that is enough to do MRI imaging. And the physiological targeting by means of the EPR effect means that they also stays in the tumor for a longer time, which means that we can reduce the background that is typically seen with today's contrast agents that are more kind of low molecular weight in form. Selective accumulation, of course, provides for better precision in MRI, which is sometimes a problem today where you get lots of false positives and sometimes false negatives in the diagnosis of tumor diseases. And the retention in the tumors also allows for more flexible imaging because you can do several MRI runs over the course of hours, and you don't need to administer the contrast agent while the patient is actually in the MRI camera. That can be done outside of the MRI room. So the main differentiation points compared to current contrast agents for MRI is, number one, is the selectivity for malignant lesions. And that is made possible by the physiological targeting with these functional particles. This in turn can lead to reduced false positives, which is a major problem, especially in breast cancer today. Where patients are frequently called back for doing second round imaging. It also reduced the MRI scanning time because the patient does not need to be in the camera as he or she is administered with this contrast agent. So this could actually allow for an increase in MRI suite productivity. And the flexibility it provides, again, we can offer the option for repeated scanning if scans are for some reason not optimal in the first round. This cannot be done with current contrast agent that basically leaves the body within minutes. So there is no need here for redosing of gadolinium agents that are used today. And speaking about gadolinium, this is not, the product we have got is based on manganese, which means there is no gadolinium involved and no kind of risk of retention safety concerns with gadolinium. It's also more environmentally friendly than gadolinium. So this was a release that was issued earlier in 2022, where we disclosed interim results. We have clear contrast enhancement, and we have good safety seen with this contrast agent in the first clinical trials. And actually, right now, we are closing the study. We have results from 14 breast cancer patients, and the trial was run in Sweden at 2 hospitals. So this is really important to us because this provides really a clinical proof of concept, not only for SN132D, but actually for the whole platform technology. We have now shown that our functional nanoparticles do, in fact, selectively accumulate in human tumors, which opens up not only for precision diagnostics, but also for precision therapy. So just looking into a little bit of a few of the images that we have seen in the trial, and I'm not going to dwell on these, but we do see clinically relevant contrast enhancement in breast tumors. And we also do see that there is much less background as you would expect from a gadolinium agent. And this selective accumulation opens up not only for, as I said, for improved diagnostic, but also for therapeutics, and also important for use in other solid tumors. So breast cancer has kind of been the model for us to go into clinical, but we are now expanding the program into other diseases and also outside of cancer with endometriosis. We also saw very nice enhancement of the pancreas and the liver, and this opens up also for better precision in imaging, since there is today no good MRI contrast agent especially for pancreas imaging, and this could be potentially very important, as you would like to, as I'm sure you're aware, find pancreatic lesions very early on, as it's really a deadly disease. So if we look a little bit into the market segment, this is kind of a static market historically. There is only one marketed contrast agent specifically indicated for MRI of breast lesions, which is Gedavist. There are a few others. There is a new one coming out that was recently FDA approved, which includes tumors in general, but the actual mechanism is not targeted, which means that you will still likely have the problems of false positives, and also it's gadolinium based, which means that it will carry the same type of safety issues that gadolinium historically have done. And there has been actually almost a handful gadolinium agents withdrawn due to regulatory concern over gadolinium. So this is something to be aware of. And more importantly, there are no pipeline products specifically targeted towards solid tumors or EPR positive lesions. So this positions SN1032D as quite unique in comparison to what it currently looks like in the market. I'm going to switch to Tumorad, our pharmaceutical project, based on the same nanomedical platform. Here, instead of manganese, we are using lutetium-177, which is a clinically validated radioisotope. It has proven to be very good in treating prostate cancers and neuroendocrine tumors and other diseases as well in the clinic. And the type of particles are similar to the one seen with SN132D, but here they are slightly bigger, which allows for a longer circulation time and a better profile when it comes to tumor targeting. Looking into the clinical concept, well, the nanoparticles are optimized for physiological targeting, so it's similar to SpagoPix. We're using a clinically effective radioisotope, lutetium-177, as mentioned. The preparation of functionalized nanoparticles is very easy. It's basically a question of mixing the isotope and the cold particles together at the radiopharmaceutical in the hospital, and then they're ready for injection into the patient. After injection, they circulate and they do accumulate in aggressive tumor and metastasis. It's been shown before that the EPR effect is more pronounced in rapidly growing tumors, which means that we are able to target more aggressive disease, basically. And tumors are irradiated on the inside. Using a radioisotope as lutetium really gives the opportunity to irradiate the whole tumor, meaning that we can kind of kill all tumor cells without the risk of leaving others viable tumor cells behind that could lead to recurrence. And then the particles are eventually eliminated through the liver. So what sets this apart from current radionuclide treatments is, again, the selectivity for malignant lesions by means of physiological targeting. This opens up for use in different types of cancers, which is different from radionuclide treatments based on antibodies or peptides and the like. Using lutetium, again, is a proven treatment concept. It's a clinically effective. Using functional nanoparticles means there is no need for drug delivery. The isotope sits with the particles and does not leave them. So the fact that the particles accumulate in the tumor is enough for us to do good. The use of this can be optimized by patient selection. Since we are using a radioisotope, we could actually leverage this as an imaging diagnostic as well, which means that there is an inherent opportunity for companion imaging, which means that we could actually use this both to follow the drug in the body, but also to select patients beforehand. Radionuclide treatment provides a very good potential for combination therapy. It's been proven scientifically that radiation can open up for improved treatment with targeted therapies like immunomodulators and such. We also have preliminary data, preclinical data, suggesting that we have a good combination effect with chemotherapy. So this is a very exciting opportunity and should definitely be explored further as we come into clinical trials. Logistics is very important when it comes to radiotherapies. We have worked a lot to facilitate this as much as possible. What we are actually delivering to the clinic is a non-radioactive kit that can be very easily radio-labeled on-site or centrally, depending on what is actually requested from the clinic, from the hospital. Many hospitals today do have the facility to do local radio labeling, which means that the drug can be freshly prepared and then brought into the patients very easily. If we look at the current pipeline and market for radionuclide therapies for solid tumors, we can see that most of the pipeline drugs and the approved drugs like Lutathera and Pluvicto are based on biological targeting by means of antibodies or peptides and beta radiation, which is what lutetium-177 is actually able to emit. There are a few compounds out there, the most prominent for perhaps Spago, which is based on alpha radiation, which has a higher energy but a much lesser reach. There has been moderate success with these drugs, I would say, but there are a few in the pipeline, and there is probably more coming out in the coming decade or so. And then we have our product, which is based on beta radiation that is clinically proven, but it's also based on physiological targeting, which means that we can target several different tumors compared to the agents that are based on biological targeting only. So with this in mind, we are looking to several different cancer indications that could be potentially targeted with Tumorad. Here we have just listed a number where we know that the EPR effect is pronounced and it has been documented clinically. As you can see, it's a mix of very large indications and smaller indications. Right now, we're very much into ovarian cancer as a potential lead indication for early clinical development, as it could provide a potential for orphan indication. We have shown preclinically that Tumorad, or the candidate drug lutetium-177 SN21, shows excellent uptake in preclinical tumors. This is a colorectal cancer model. It does inhibit tumor growth and extend survival, not only in this model, but also in others. And we also know from our imaging diagnostic that our particles do accumulate in tumors in the clinic in human cancer patients. So with this in mind, we are now preparing and focusing very much to prepare the first human clinical trial with Tumorad. It will be a phase 1/2A trial, where the phase 1 part is expected to start in the spring. The main purpose will be to study the safety and tolerability, and more importantly, to find a dose that could be suitable to treat patients in the phase 2 part. We do have financing for the phase 1 part here, but we are looking as we speak for financing for the phase 2A part of this trial. And looking a little bit more into the kind of broader segment of radiopharmaceuticals, we can see that there is an increasing interest in this field over the past, let's say, 4 to 5 years. And it's I think, to my mind, very much driven by the successes of Novartis and with lutetium-based drugs in different indications. If we look into valuation and using only ovarian cancer, which is one rather small indication, we see that the valuation right now can be probably very much higher as development go along, and the targeted market in the end is very large also when it comes to this small indication of ovarian cancer. So this is just an example of the magnitude of this drug if it's successful in the clinic. So just to summarize, we have a solid platform to address major medical needs. We have a proprietary technology. It's owned by us in full. We have a good patent protection for both projects. We do have clinical proof of concept of the mechanism and of the particles by MRI in breast cancer patients. The preclinical efficacy in different cancer models looks good, both as monotherapy and in combinations. The CMC is in place. We have an established process for GMP production and IMP release. Excuse me. And there is also imaging biomarker options for companion diagnostics and/or a theranostics approach if that's something that would be considered. And we have a development plan to market in large indications and also that could be optimized by an orphan drug strategy. So I think we're quite well set with this, and I should also conclude to say that we're a listed company. We have an experienced team in the management and in the board with many years of both preclinical, clinical, marketing, business development, and financing experience. So with this, I thank you for your attention, and I'll be happy to discuss this more in detail with any one of you. Thank you.